Hello guys and welcome back to Social Academy. Sorry for the delays which is happening now. I'm not able to upload the videos, but I'm trying my best to complete these. Today we are going to talk about the oncogene targeted drugs. So already guys we have discussed in the previous lecture that once you have got the knowledge about the oncogene and the tumor suppressor genes, right? So we know that the gain of function, that is the activation of the oncogene and the suppression of the tumor suppressor gene and all. If we know all these things, we can actually go for the personalized drug therapy. What does it mean? It means each cancer patient is going to have its own therapy regimen. Okay, so now this is possible. So it is not like in a mass that, suppose we have got a group of liver cancer patients that we have to give them all the same drug. It is not going to be like that anymore. So now what we can do is based on the stages of the cancer and the biomarkers which we have found and the amount of the mutation happened to the patient, the drugs can be designed and it can be added in the therapy regimen. Okay. So in this lecture, we are going to discuss how oncogenes can be targeted for the drug therapy. Okay, so a critical question is whether the discovery of oncogenes and the tumor suppressor genes will allow the development of new drugs that act selectively against the cancer cells. So guys, आज के lecture में हम ये देखने वाले हैं कि कैसे oncogenes और tumor suppressor genes को हम target करके drugs बना सकते हैं. इसमें हम ये भी देखेंगे कि it is not always possible to do such things because we know that these proto-oncogenes and the tumor suppressor genes, they are also part of the cell cycle regulations and the different aspects of the cell signaling. So अगर हम उसको टारगेट करेंगे, so eventually we are also affecting the normal cell development. तो इसलिए drug therapy इतना आसान नहीं है, ठीक है? तो यही सब हम इस lecture में आज समझेंगे. So most of the drugs traditionally used in cancer treatment either damage DNA or inhibit the DNA replications. Consequently, these drugs are toxic not only to the cancer cells but also to the normal cells. So what is happening is particularly those normal cells that are continuously replaced by the division of stem cells. The action of anti-cancer drugs against these normal cell populations, this will say kya hoga? It is going to be toxic and eventually those normal stem cells are also being, they are also dead, and because of which what will happen? आपने देखा ही होगा कि cancer patients के ऐसे loss of hair पहले हो सकता है, ठीक है? So that is like one of the signs that the chemotherapy is okay. A promising strategy for achieving more selective cancer treatment is the development of drugs which is being targeted against the oncogenes that derive the tumor growth. Unfortunately, from the standpoint of cancer treatment, oncogenes are not unique to tumor cells. This is what I was telling you. So these oncogenes basically क्या है? आपके proto oncogenes जो है हमारे जीनों में present है, ठीक है? So they are the part of normal cell signaling and development proliferation structures, right? So when there is a mutation happening to these proto oncogenes, then they are getting converted to the oncogene which is responsible for the cancer. अभी अगर मैं इसको टारगेट करके ड्रग बनाऊंगी, so actually I'm actually also disturbing the cell signaling pathway. So जिसकी वजह से क्या होगा? Normal signaling जो है वो भी affect हो जाएगा. So the exploitation of oncogenes as targets for anti-cancer drugs is therefore not a straightforward proposition, but we can have the selective oncogene targeted therapies. For example, already in the market में है, but of course they don't give the guarantee that it is going to cure the cancer. It can just increase your survival rate percentage. For example, Herceptin is to breast cancer, to again colorectal cancer. So हम यहाँ पे देखेंगे कितने सारे जो हैं, it is a targeted protein like ERB2 again and again. So here हमने breast cancer, colorectal cancer and lung cancer में हम use कर रहे हैं. So ठीक है. तो अब ये सवाल आता है कि कैसे it is becoming specific for the lung and breast and colorectal cancer. No, it is not that. It is not becoming specific. It's just that particular protein, if suppose targeted, it is just stopping the proliferation of the cell, and that's how the breast and the colorectal cancer or the lung cancer is getting inhibited. ठीक है? तो ये थोड़े से examples हो गए हैं. Just you can guys go through it. सिर्फ Herceptin भी याद किया, Imatinib भी याद किया तो काफी है. Okay. The first drug developed against a specific oncogene that was approved for the clinical use was Herceptin, which is a monoclonal antibody against the ERB2 oncogene oncogene protein. Now it is overexpressed in the 25 to 30% of breast cancer because this protein is getting overexpressed, amplification. Okay? So it was first found that the antibody against the extracellular domain of ERF2, that is a receptor tyrosine kinase, it was inhibited the proliferation of tumor cells. जहाँ पे भी हमें इसका overexpression मिला, तो इस हिसाब से हमने उसको ड्रग के रूप में उसको मार्केट में लाया. Now these results led to the development and clinical testing of Herceptin, and बहुत सारे women made clinical trials and they found that whenever there is overexpression of the ERF2 protein, breast cancer है, और जब आपने Herceptin दिया तो there was the delay. That is, there was the good clinical response of the therapy response, which was seen in the case of the women which were under the treatment. So finally, it was given the approval by the FDA in 2001. So the therapeutic use of monoclonal antibodies is limited to the extracellular targets such as the cell surface receptors. अब इसका क्या मतलब है? It means that if suppose this is your cancer cell, here you have got the cell surface receptors, and to this the monoclonal will come and bind, and this will induce the apoptosis or some other pathways which is resulting in the death of the cancer cells. A more widely applicable area of drug development is the identification of small molecule inhibitors of oncogene proteins, including the protein kinases that play key roles in signaling the proliferation and survival of the cancer cells. Again, the pioneering advance in this area was the development of selective inhibitor of the BCR avial tyrosine kinase, which is generated by the Philadelphia chromosome translocation, which is seen in the case of chronic myeloid CML. Guys, please, ये जो ऐसे छोटे-छोटे चीजें हैं आपको हमेशा याद रखने लगोगे Philadelphia chromosome. ठीक है? किसके केस में देखा जाता है? CML क्या है? Chronic myeloid leukemia. So guys, all these structured forms are important for you to remember. Now let's see this. So what happens in the Philadelphia chromosomes? So you have got the two chromosomes, 9 and 22. So we have got the gene over here, ABL and BCR. And because of the translocation, what happens is that the BCR and ABL comes together. Now this fused chromosome is known as the Philadelphia chromosome. आप यहाँ पे भी देख सकते हैं किस तरह से translocation हुआ है, ठीक है? So Brian Drucker and his colleagues developed a potent and specific inhibitor of this fused case, that is BCR-ABL protein kinase, and it showed that this compound blocks the proliferation of the CML cells. So what will happen? There was no chronic myeloid leukemia, यानी कि यहाँ पे unregulated proliferation of cells नहीं हो रहा था, ठीक है? So once you get all these results, a clinical trial of this particular drug was done, and वो drug कौन सा आपका Imatinib. मैंने इसलिए आपको बोला कि ये दो जो drugs हैं, Herceptin and Imatinib, please remember because the questions might be coming from these two. And then we got very good, remarkable results from the usage of Imatinib. So guys, एक और चीज यहाँ पे ध्यान रखना, हम हमेशा यही बोलेंगे that it has showed the good effective therapy response, but we can never claim that it has 100% cure the cancer. You can never say that, even though the patient has recovered, but there is a chance that after 1 or 2 years there will be a relapse. A relapse मतलब क्या हुआ? Again the start or the onset of the cancer. So the striking success we saw in the case of the again it was improved by the FDA in 2001, and then we saw that there was a decreased mortality by using this particular drug, that is your Imatinib. ठीक है? तो इस तरह से एक चीज और आपको समझना होगा यहाँ पे that whenever from the lab you see that this particular Imatinib is showing very good response in the in vitro cell lines, then you go for the in vitro हो गया आपका पहले, उसके बाद आप जाओगे animal cell lines, animal studies में जाओगे, and once you get over here also good results, then you can go for the, you can ask the FDA to go for the clinical trials. ठीक है? And once you complete the clinical trials, then only the drug comes into the market. Okay, so ये drug का नाम हो जाएगा इसको और इसको market में आएगा. So don't worry guys, I'll take a separate lecture of this clinical trials. Okay. So this is the incidence, the effect of Imatinib on mortality from chronic myeloid leukemia. So इतने incidence हैं और उतने आपकी mortality rate है. So what you see over here is once you start using the Imatinib, ठीक है, 1997 में शुरू हुआ, there was decrease in the mortality rate. You can see over here. Over a period of time, the rate of the CML was almost the same, like between 2,000 to 500,000 patients were there. But the rate of mortality, यानी कि लोग जितने मर रहे थे, उनका number गिरता चला गया. So that shows that Imatinib has shown very good effective of the therapy response towards this particular CML. Okay. So and the mortality rate has been decreased. So इसलिए फिर FDA has approved this drug to be given to the market. So by analysis of these resistant mutants, it has been possible to design new inhibitors, which are currently being tested in clinical trials against the leukemias that have become resistant to Imatinib. So कैसा होता है over period of time कोई भी ड्रग हो, फॉर एग्जांपल अगर आप 10 साल से पैरासिटामोल ले रहे हो, there will be a time when your body will become resistant to the effects of the paracetamol. Then you need to increase either the dose of the paracetamol or you need to change the drug. The same way, the cancer cells also become resistant to the Imatinib. For example, over a period of time, then what happens is that the scientists, the doctors, they have to change the therapy regimen. Okay, यहाँ पे आपको drug को change करना पड़ेगा because the cells have become resistant over a period of time. So you need to change the drug. तो इसी हिसाब से कैसे रहता है? There will be more mutations which is happening to the CML, and then you need to design and redesign the drug so that it becomes more effective against the CML. So it is also potent inhibitor of the PDGF receptor and the KIT tyrosine kinases. It has been proven to be effective therapy for tumors. Okay. Now KIT is activated as an oncogene by point mutation, resulting in constitutive protein kinase activity, which is 90% in the case of the gastrointestinal stromal tumors. Okay. Now, many of the gastrointestinal stromal tumors that do not have activating mutations of the KIT, they have mutations of the PDGF receptor. So, अगर एक नहीं तो दूसरा तो होता ही. So these are highly responsive to inhibitors. Otherwise, कैसा रहता है? First is we thought that Imatinib is specific only for the CML, chronic myeloid leukemia. But later on, they also they also observed that there were certain the effectiveness was shown against the other kind of cancers like gastrointestinal. इस तरह से हमें पता चला है कि the same drug, suppose once it becomes resistant to the CML, but it can be used against the gastrointestinal stromal tumor, then it can be again used and it will be there in the market. So ये कैसे change हो रहा है, कैसे पता चलेगा? ये every time the research has to be conducted for further study. So in addition, Imatinib is active against other types of tumors in which the PDGF receptor is activated as an oncogene, including the CML, in which it is activated by fusion with the cell transcription. So आप यहाँ पे देख सकते हैं, ये मैंने मेरे पहले lectures में भी discuss किया हुआ है. So normally what happens is that the PDGF comes and binds its receptor. Then what happens is that the domain over here, the kinase domain gets activated and they self-phosphorylate. ठीक है? अब यहाँ पे क्या हो रहा है? In this case, what happens is that the receptor is expressed in such a way that it is acting that the receptors are present. It is acting as if the ligands are binding to the receptor. So the moment it is expressed on the plasma membrane, they are also highly active. ठीक है? इस तरह से ये जो signal है आपका active रहता है. So it becomes constitutive. So two small molecule inhibitors of the EGF receptor, that is EGFR receptor का inhibitor जो हो गया, आपका Gefitinib हो गया, Erlotinib हो गया. They have shown striking activity against the subset of lung cancers in which the receptor is activated by the point mutation. So एक example हो गया जहाँ पे where you can see that the TEL PDGFR oncogene activation is there. So what drugs can you use? You can use Gefitinib and the Erlotinib. Don't worry guys, ये आपको पूछेगा नहीं, this is only for your knowledge. So this is how so many mutations which is happening in the oncogene. So there are many inhibitors also which has been studied in the clinical trials in the labs and all are also present in the market. Similarly, you have got an inhibitor of the B-Raf also, which has shown striking results in the treatment of therapy. So guys, ऐसे आप देखोगे कि इस तरह से काफी cancers हैं जिसका against drugs भी हैं, which is available in the market. They are approved by the FDA. So whenever you take any medicine, you need to see that these drugs are approved by the FDA. तो ये बात होगी overall world की जहाँ पे FDA approval होता है. So there is one question, guys. I'm asking that if you're watching this video, this question is meant to benefit you. So which is the regulatory authority in India for drug approval. Okay. जैसे सभी के लिए FDA हो गया है, यूएस में FDA अप्रूवल चाहिए होता है, वर्ल्ड वाइड, वैसे ही इंडिया में अगर किसी ने आपका ड्रग खाया, ठीक है, we also have so many companies, so many clinical research organizations are there. What if we also have so many pharma companies? What if we are making or synthesizing a drug which is giving 100% or 90% effectiveness? Okay, so what is that regulatory authority in India which is going to give the approval for the drug to be used into the or introduced into the market? So you guys have a question here. If you're watching this video and listening to me, this is a question for you. अगर आपको इसका आंसर मिलता है, good. If not, then please let me know so that I can give the answer. Okay, so आगे बढ़ते हैं. So then other oncogene targeted drugs have been made to the ligand PDGF kinase. So आप एक चीज समझ लीजिए, जो भी आपकी सेल सिग्नलिंग की important proteins है, so many drugs have been made. एक चीज मैं आपको बताती हूँ, फॉर एग्जाम्पल लैब में अगर सपोज एक रिसेप्टर है, यहाँ पे आपका लिगेंड आ रहा है, ठीक है, now this is activating one protein, this is activating another downstream protein, this is activating third, and then it goes into the nucleus and then having the gene expression, ठीक है. So and there are 10 labs which is working on the same pathway. So what they do is like, and this is nothing wrong also, because you never know which one will hit the jackpot. You never know which will be the drug in the market which is making you the millionaire. So either what you can do is inhibit the receptor so that the ligand doesn't bind over here. Okay, you can modify, you can make an inhibitor to the receptor. You can also make an inhibitor of this particular protein, this particular protein, this particular protein, this one and this one. So इस तरह से भी आप कर सकते हो, ठीक है. So there are many ways by which you can actually stop or inhibit that particular cell signaling pathway. So एक signaling pathway and you can get 10 to 20 candidates for the drug design. So एक तरीके का भी research हो गया है, which is actually good, and you never know which one will be in the market accordingly. Okay, this is another pathway. Again, you can see C-growth factor comes, it binds to the receptor, it gets autophosphorylate, it's activated, then PI3 kinase has got the ancestry domain, which binds to the phosphorylase, the tyrosine kinase domain. This gets activated, it is going to activate or convert from PIP3 into PIP3, which then gets bind to the mTOR-C2, which activates your AKT. AKT gets activated and then it is going to for the further transcription or inhibition of the effectiveness of oncogene inhibitors in cancer treatment. Suppose the hypothesis is that tumors with activated oncogenes are particularly susceptible to the inhibitors of these oncogenes. Effectiveness of oncogene inhibitors. It's suppose a hypothesis that tumors having the oncogene activation are susceptible to inhibitors of those oncogenes. Okay? So this is also consistent and shown in the case of the animal model also, and we have also seen where they have tested in the secondary signaling pathways. Okay. Consequently, the proliferation and survival of the tumor cell may become dependent on continuing activity of the oncogene, whereas normal cells have got the alternate pathways. Okay. A cancer cell with an activated oncogene may therefore be selectively sensitive to inhibition of that oncogene encoded protein, and that's how the drugs are actually affecting the cancer cells. So this is the ways by which you can actually make that. Now, अभी हमने ये देखा, even though there are many drugs which are targeted against the oncogene protein, still we are not able to cure the cancer, right? Because many drugs are getting the resistance to, the cancers are getting resistant to these drugs. Okay, so कैसे होता है? For example, like you have got the oncogene activation of RAS or PI3 kinase, which can result in the resistance to the EGF receptor inhibitors. So over period of time, what happens is that, जैसे मैंने आपको बोला कि like in 5-10 years, the particular drug or the particular cancer cell lines, cancer cells are becoming resistant to that particular drug. Now the question is coming, why? Okay. this can be your second assignment. This is really very simple, like why over the period of time this resistance is coming, and the answer is very simple. This question we have asked in the interviews also many times. This is like your interview questions. So guys, if you're listening, let me know the answer of this question also. This is really very, very simple. And if you don't know this answer, then really you need to brush up a lot your basics. Okay. So even though we have done so much of research, we came to know that there are many drugs which are happening out, which are showing promising results against the cancer cells. Still, many more research has to be done because हमसे ज़्यादा cancer cells are more brainy and hardworking. So we do take time to rest and all, but the cancer cells keep on dividing and they all keep on mutating. And these unregulated mutations are actually making our job difficult in curing those cells. Okay. तो इस तरह से कैंसर सेल लाइन जो कैंसर स्टडी है those research doors are always open. You guys can also jump into the cancer, like how I did. I always wanted to work in cancer field, and now of course I'm working in that only. So I'll be sharing about my experience also, like how I jumped into the cancer biology and how it has given me the opportunity where I can show and I can show lots of thinking, and I can use my knowledge for the benefit of the society, etc. So till then, guys, please subscribe my channel, let your friends also know, because at the end of the day, only target should be clearing CSA errors. अगर आप बायोलॉजी में हो, अगर आपने MSc बायोटेक माइक्रोबायोलॉजी केमिस्ट्री किया है, कोई बोले कोई ना बोले, I am always going to make sure that say this, that clearing CSR is very important. If you really want to make the academics, yes, that is like it should be one of your major goal right now. अगर आपने कर लिया है, good, congratulations on that. But if not, please work hard, study hard, and clear the exam. So तब तक के लिए guys, take care.